All right. Any questions from last time? If I asked you what a constraint would be, could you give me an example? Okay, what specifically would be an energy constraint?
Reproduction or survival. Like you put energy towards.
Okay. Yep, okay, yep. So energy that you put towards growth and maintenance is not energy that you would put towards, that you could put towards reproduction, because you can't double dip on energy in that way. Okay. An example of a genetic constraint.
The snakes that we talked about.
Okay, snakes.
Stripes versus the checkered.
Okay, so in that case, we looked at stripedness and reversals. Those would be genetic constraints, because those two things have become negatively genetically correlated with one another over evolutionary time, because of the way in which the color pattern is tied to the behavior in terms of who survives and who doesn't. So that would also be a genetic constraint because of that negative genetic correlation. If you had a positive genetic correlation between two traits, that's not a constraint, because the correlation isn't negative. So, all right. So the last thing we want to talk about before the exam is about testing hypotheses in behavior. Once again, they talk about this in Chapter 2 of your textbook, and we'll actually use some of their examples. When we go to evaluate hypotheses in animal behavior, and this is true to a lot of biology, but it's especially true of animal behavior, we have the choice of looking at comparisons among individuals within a single species. So we could look at how Mason differs from how Avery differs from how Elizabeth differs from how Emily differs, and see how those individuals are behaving differently, and look at variation among individuals within a single species. We can do this sometimes by experimenting. We've already looked at some research that has been done where experimentation was done on individuals within a species. And then we can also look at comparison among species. So different species do things differently. They behave differently. And oftentimes we can glean information about the evolution of behavior by looking at human beings versus gorillas versus orangutans versus macaques, etc. All of those different species do things differently, and especially if we know what the phylogenetic relationships are, the evolutionary relationships among these different groups are, we can use that information as well to figure out how behaviors have evolved, what the ecological drivers are that are leading to different behaviors in different species. So the example that we've looked at so far, we read the paper by Joseph and Pinchot looking at shrike behavior. In this case, which of these three methods was being used?
Between individuals within a species.
So they were looking at individuals within a species. What else was being done?
They were also experimenting.
They were also using experimentation. So simply because you have these three things doesn't mean that you can't use them in concert with one another, but they're different ways of going about things. So in this case, they had specific hypotheses about this behavior, impaling behavior by shrikes, and they made predictions, but all these predictions, for the most part, are about individuals, except for the first one. The first one is about the population as a whole. Concentrations of impaled prey in prominent places should be greatest at the onset of breeding season. That's when individual females would be evaluating individual males. When females arrive, males with richer caches will pair before those with poorer caches. That's an individual kind of thing. Some birds have big caches, other birds have small caches. Those differences in individual behavior should result in differences in the behavior of females. Males with bigger caches will beget more offspring. Once again, these individual differences within a species have an effect on the fitness of the individuals. And in this case, they evaluated these hypotheses using an experimental paradigm. But they could have just as easily used an observational paradigm where they just quantified the caches and then looked for correlations between cache size and date of pairing, number of offspring, et cetera, et cetera. It wouldn't have been as strong of a test of the idea, but it would have given you an answer, just not as strong of an answer as you would have if you had done an experiment. So you're already familiar with methods that look at individuals and use experimentation. You've been doing this all 4 years you've been here, or all 3 years you've been here. You're probably not familiar with what's known as the comparative method, so that's what I want to spend most of our time today talking about. This looks at differences in behavior across species, across genera, across families in some cases. And so there are specific methods by which we do this. So one example in your textbook is looking at the breeding behavior of gulls. We have different species of gulls that do different things. Black-headed gulls are ground nesters. Kittiwakes are cliff nesters. And so do you lay your eggs out on a beach where anybody can get to them? Or do you lay them up on a cliff where it's very hard for predators to get to them? So in both these cases, one of the ecological drivers that may be affecting behavior is the presence of egg predators. If you lay your eggs on a cliff, you don't have to worry about that. If you lay your eggs on a beach, you do have to worry about that. Well, when you look at traits related to reproduction, you see that you have nest site on the ground versus on the ledge. Predation risk is higher on the ground than it is in cliffs. Adults, how do they respond to predators? Well, in black-headed gulls, they take flight early, they have alarm calls, and they will attack the predator. With kittiwakes that are on the cliffs, they stay on the nest until the predator is close. They rarely have alarm calls. They don't put up much of a fight at all. So in an environment where predation risk is high, the parents are active in terms of defending the nest. In situations where predator risk is low, they haven't evolved the same strategies to fend off an attack when an attack occurs. Nest construction, not particularly elaborate. In the case of cliffs, it's much more elaborate, and there's a deep cup that cradles the eggs. Why would a cliff nester want that? Yeah, you don't want your egg rolling off the cliff. In the case of a black-headed gull, if an egg rolls away from the nest, it's only going to roll a few feet away, and then the parent will just corral it back into the nest. It's not going to, roll 30 meters down to the ocean, and it's not going to fall after rolling a couple of inches and fall to its death at the bottom of the cliff. In terms of nest concealment, the adults don't defecate near their nests. Bird feces are oftentimes bright white, and so defecating near your nest advertises the presence of the nest, and so they fly away and defecate someplace else and come back to their nest. And then also the adults remove the eggshells. Once again, these eggs, the inside of the eggshell in particular is bright white. Oftentimes, if you're a ground nester, the outside will be camouflaged. And so after a hatchling hatches, they'll remove those old eggshells because that bright white inner surface of the egg is showing, which once again advertises to a potential predator that there's a nest there. In the case of the kittiwake on the cliffs, they defecate near the nest because once again, predation risk is relatively low. They don't have to worry about that. And they don't remove the eggshells. Chick behavior. The ones on the ground are cryptic. They have weak claws. They leave the nest after a few days. They're more precocial rather than altricial. They run away when they're attacked. They have wing flapping and jumping during their development. Kittiwakes, not cryptic. They're white and gray. They ignore disturbance. They have strong claws and muscles for clinging to the cliff face. They remain in the nest until they can fly. So they're not very precocial. They're more altricial. They don't run off when a predator comes because running off means falling off the cliff to your death if you can't fly. And they're generally less vigorous, probably because they don't have a lot of space up on this narrow ledge. Recognition of parents is relatively quick in black-headed gulls. It takes more time with the kittiwakes. Once again, because if you're nesting on a beach with a bunch of other black-headed gulls, there's probably a value that has been, imposed upon these animals over evolutionary time in recognizing your parents relatively early on. If you're on a ledge on a steep cliff, probably the only birds you're coming into contact with are your siblings and your parents. So there's not as much of a need to do that. Chick feeding, parents give food calls to attract the young because they might be off hiding. Hiding from who? Hiding from predators. No parental food calls because your chicks are on your ledge, because that's your ledge and that's where your chicks are supposed to be. The parents regurgitate the food onto the ground, creating a space that they come and eat, as opposed to adults passing food directly to the young's bill. Once again, that's probably just a spatial constraint due to the nest laying area. So here you have these two different gull species that lay their eggs in two different places, and as a result of that, there's all this correlated change in everything about the reproductive system and the development of the babies and the behavior of the babies, et cetera, et cetera. So you have an ecological driver that drives one set of animals to behave one way and a different set of animals to behave another way because of that initial ecological driver, which is the risk of predation that varies by the kind of nest site that you choose. So this is one way of looking at comparative data and making inferences about differences in behavior by looking at what different species do. It's not a very formal method of comparative analysis. We can look at primates. Clutton, Brock, and Harvey, this is once again an example in your textbook. They quantified ecological data and behavioral data. They considered a bunch of alternative hypotheses. They used multivariate statistics to examine the effects of each ecological variable on behavior. And then they used generic level variation to make the data more phylogenetically independent. And I'm going to talk about phylogenetic independence here in a minute. But you can look at things like testis weight and body weight. And so obviously larger primates have larger testes simply because they are larger. They also have larger heads and longer arms, et cetera, et cetera. But when we think about testis size might be important in determining what type of breeding system a particular species engages in. So breeding systems are things like monogamy, polygamy, et cetera, et cetera. So you can be monogamous, which means that you either mate with one partner, either for a single breeding season, or in some cases, individuals will form what we call lifetime pair bonds, where you're stuck with that mate for the rest of your life. Humans are sometimes referred to as monogamous, although when you look at the data, we're considerably less monogamous than we might like to think of ourselves. In this country in particular, we have a high divorce rate. That is not a sign of monogamy. In some cases, individuals are polygynous or polyandrous, depending on the individual. Do you guys know what all these words mean? Yeah, okay. So monogamy one male, one female. Polygamy is a more general term, which could mean either one male, many females, which would be polygyny, or polyandry, which is many males and one female. Interesting how we as humans talk about polygyny and polyandry if you see discussions of this type of mating system in humans, we almost always refer to it as polygamy, in spite of the fact that in almost all cases, not all cases, but in the vast majority of cases, when we have one individual with many partners, it is almost always, not absolutely, but the vast majority, it's almost always this. It's almost always polygyny, one male with many females. It does occur, but to a lesser degree, that we have one female with many males, but once again, that is a less common occurrence in humans. Yet we tend to use this more general term, polygamy, when we talk about polygyny. The most common place in which we talk about polygyny is when we talk about bigamy, which is almost always used to define one man with two wives. And once again, this is just a weird social, cultural, sexist way of talking about mating systems in humans. I would much prefer that we use the more specific terms when we talk about any specific non-monogamous relationship in humans. Now, monogamy can either be lifelong pair bonds, or we can also have serial monogamy. Serial monogamy is when you have one male and one female pairing for a season of reproduction, and then the next breeding season you may have that same male pair with that same female again, or you might have that male pair with another female. It doesn't really matter, but the monogamy is of short duration for a given season. And once again, the monogamy that we talk about in humans could be most best described as a form of serial monogamy, once again, given behavior of humans. You are monogamous for a time until you get a divorce, and then you go and be monogamous with somebody else. Or you're monogamous with somebody without getting married, and then you break up and you go and be monogamous with someone else. So, when we look at test size in the breeding season across a wide range of primates, one of the things that we see is that when we plot the breeding system on this plot, a pattern emerges. The open circles are monogamous in monogamous species. You see that they're all very close to the line or slightly below the line. Open triangles are single male systems, one male with multiple females. The closed circles are multi-male breeding systems, a situation where you have lots of males competing for the available pool of females. And so one of the things that you find is that if you are a male competing with a group of males in your social group for a group of females, that tends to correlate with a larger testis size. All sorts of evolutionary reasons for that. One of them could be sperm competition. If larger testes give you more sperm total in terms of your total sperm count, if you have multiple males breeding with multiple females and multiple females breeding with multiple males, your ability to reproduce may just be correlated with your ability to produce more sperm than the other guy. And so we see this pattern where testis size as it relates to body size, there's a pattern that correlates with the type of breeding system that you happen to be in. Now, this is done at the generic level, and their attempt to get rid of phylogenetic effects was by looking at generic level data rather than species level data. So that's one form of the comparative method. Let's go to this. So what we see when we do the comparative method is that here's the phylogeny of primates. They arose here, two major groups. Homo is right here. This is human beings. These are the chimpanzees, gorilla, the pongo. Why cannot not remember that? Well, I don't remember what genus pongo is.
Orangutans.
Oh, orangutans. Thank you. Did you just look that up? Okay, cool. All right. So these are what we oftentimes refer to as the great apes, the large apes. They are a phylogenetically distinct group from the rest of the old world primates. And so, if there is a particular trait that arose in the common ancestor of the great apes, it might be that all the members of the great apes have that particular characteristic because the common ancestor had it. It's possible that chimpanzees and humans both use tools, not because chimpanzees developed using tools independently of humans using tools, but because we came from a previous ancestor, and a common ancestor that used tools, and as a result, both of those groups have the ability to use and craft tools because we inherited it from a common ancestor. And so when we talk about phylogenetic independence, we have to separate the degree to which you possess a particular characteristic because it evolved independently in your lineage versus you possess that characteristic because you inherited it from a common ancestor, which means that you might expect to see that same character in other taxa. So you want a way of analyzing those characteristics in a way that takes phylogeny into account. And so what we've developed, what we biologists have developed, I didn't do it, a bunch of people back in the 70s and 80s did it, is we use this technique called constructing phylogenetic independent contrasts, and I'll explain to you what that is about. So species may share similar characteristics because of convergent evolution, meaning that one group of unrelated organisms does things one way and another group of unrelated organisms does it a similar way, not because they share common ancestry, but because they share common ecological circumstances. And so those selection pressures imposed by the environment led to common solutions to a common problem. It is not inherited from a common ancestor. But you can also have inheritance from a common ancestor due to these phylogenetic relationships, and it would be nice to be able to tease those things apart. The only way we can tease those things apart, though, is by having a phylogeny. So how do phylogenetically independent contrasts work? If you have a phylogeny, rather than dealing with the raw data themselves, what you do is you say, if this is the value for trait 1 in taxon D and the value for trait 2 in taxon E, trait 1 in D and E, trait 2 in D and E, and you're interested in how trait 1, or X, correlates with trait Y, you could just use the raw data, 20, 20, and 120, or 16 and 110, the correlation between Y and X. You can do the same thing here. You can figure out what the common ancestors are based on the phylogeny that you have, what their values would be. In some cases, we have fossil evidence where we can actually measure that directly, but there are algorithms by which we can figure out what this is. As it turns out, when doing comparative methods, trait X in ancestor B is simply the average of the trait in D and E. So 20 and 16 gives you 18, 120 and 110. gives you 115. So what you do when you do this is you're looking at what the That's weird. Those aren't the right numbers. Oh, there it is. Oh, yeah, I gotcha. What's the difference here? 20 and 16, the difference is 4 for x. The difference is 10 for y, which is right there. So it is slightly off. But you plot these contrasts. You see how they differ from one another, and then you plot that difference. And you plot that difference for sister taxa. So you take taxon D and taxon E. What's the difference for trait X? What's the difference for trait Y? And you plot that. You look at F and G. This is contrast 2. 4 and 6, they differ by 2. 93 and 87 differ by 6. So 2 and 6 would be right there. And then the common ancestors is contrast 3. 18 and 5 is 13, which is right there. And 115 to 90, that's 90, 100, that's 25. And so that's where that one appears. And you do this across the entire phylogeny. So if we were going back to these guys, you could do independent contrast between pan and homo, and then between the common ancestor of pan and homo and the gorilla, and then between the common ancestor of pan, homo, and gorilla with the pongo. And at each of those points, there would be a contrast between those. And at each point, you're getting further and further away from the original data, the original characters of the actual individual taxa. You're dealing with how do those taxa differ with one another, and does the change in one trait correlate with the change in another trait across different levels of this phylogeny? That then removes, to a certain degree, the fact that all of these individuals are related to one another. And so, while you may be able to do a correlation between trait 1 and trait 2, trait Y and trait X, for all these individual taxa, and you might find that that correlation is strong, it's positive and strong, like there's a good degree to which variation in trait X explains variation in trait Y, but that could just be an artifact due to the fact that maybe these things all differ in size, and those two characteristics are also correlated with size. But we can remove those kinds of things by seeing how trait 1 changes relative to how trait 2 changes across the phylogeny. And so that's what this is doing, this is attempting to do. So that when we do then see this correlation in the contrast, we can now say more definitively that these two things are correlated, and they're correlated independent of phylogeny. So it's a little difficult to understand, so I'm going to take you through an actual example. So I have some programming skills in R, and there's a program in R that allows you to do phylogenetically independent contrasts. So what I did was I went into R, and I told R to build a phylogeny. And I told it to build a phylogeny where you have two groups of related individuals, and these two groups of related taxa are separated by a really long divergence that separates these two. So time is on the x-axis, and these two groups evolved a long time ago, but they separated early in evolution, and then recently in evolution they went through an adaptive radiation, and then another adaptive radiation down here. And then what I asked R to do is I asked it to do what we call a random walk of evolution of two characteristics. And what a random walk is it basically just alters the phenotype for a particular trait a little bit above where it is now or below where it's now, and it's random. And so you get a trajectory of phenotype that looks like this, where the phenotype varies, but if it starts trending in a particular way, it might be more prone to continue trending that way. If it's trending in a particular way, it might tend to trend more that way. And I had R do a random walk of evolution for 2 traits along the entire phylogeny, this phylogeny here. So that at the end of this, what we ended up with is trait values for trait 1 and trait 2 for every twig on this phylogenetic tree. Once I had that, I then did a correlation, said, to what degree is trait Y correlated with trait X as a result of random evolution? And what we find is that those 2 things are correlated with one another. And so we might say, through random evolution on this evolutionary tree, there is a correlation between X and Y. But there's really no reason for there to be a correlation between X and Y, because the evolution of trait X was random and it was independent of the evolution of trait Y. So how did these 2 things become correlated with one another? As trait X gets bigger, trait Y gets smaller. Well, if we impose that original phylogeny on this scatter plot, we find that this line in the middle here is actually that long divergence. And this is one group of the adaptive radiation. This is the other adaptive radiation. So what happens is all of these taxa have similar phenotypes because they share a common evolutionary history. All of these taxa have a common phenotype or close phenotypes because they all share this long evolutionary history. So what we have is this group is the upper group, and this group is the lower group. And so what you really have isn't a negative correlation between lots of different taxa. You have a negative correlation between this branch of the evolutionary tree and this other branch of the evolutionary tree. So how can we fix that? Well, we can go through and look at these 2 taxa, say how do they differ for trait X and Y? Look at these 2 taxa, how do they differ in X and Y? Look at this taxon, this ancestor versus this ancestor. How do they differ in X and Y? We can calculate these independent contrasts. Once again, we can do that on this data set, and what we find is that there's no correlation whatsoever. These are the correlations between the contrasts, the phylogenetically independent contrasts. And that correlation that looked significant before, we see was really just to the effect of phylogeny, because when we look at the correlation that has had phylogeny removed from it, we see that it's just a random scatter point of X and Y. X is not evolving in conjunction with Y at all. Well, of course it isn't, because I set it up so that they didn't evolve in conjunction with one another. I set it up so that trait X was evolving by itself randomly. Trait Y was evolving by itself randomly. What made them look like they were evolving in conjunction is because trait X and Y were evolving along this phylogeny. Trait X and Y were evolving along this phylogeny. That then made them look like they were negatively correlated, when in actuality, they're not. They were random, because that's how I set it up to be. And so that's the advantage of phylogenetically independent contrast. It gives you an estimate of whether or not 2 traits are correlated, while controlling for the fact that these organisms are all related to one another. So we can hopefully see what is the truth rather than something that is skewed by shared evolutionary history. What I did was this thing here, just on a much larger scale in terms of the size of the phylogeny.
So what kind of question would you ask on the exam like that? Like what?
Well, this is one of the hardest questions to ask on an exam because it's more about a concept and a process than like a right or a wrong answer. So how would I ask this on a test? That's a very good question, which I should figure out between now and Friday morning. So one of the things that I might do, here's something that I could do, and it would be a fair thing to do, is I could say, a researcher has a hypothesis that the cache size is related to the level of polygyny. So cache size, so strikes are not the only things that build caches. We build caches. Our caches in many cases are caches of money. In some cases our caches are caches of food. Walk into any house, there's a pantry. Some people's pantries are bigger and more complicated than other people's pantries. And even within people with similarly sized spaces, some people cram those pantries full of stuff and other people have much smaller and as a result much more orderly pantries because they organize them. And so they tend to stockpile food for shorter periods of time rather than longer periods of time. The difference between a prepper and a normal person, for example. You guys know what preppers are? People preparing for an apocalypse and they have the root cellars crammed full of canned goods and munitions and everything else. So lots of organisms cache. Squirrels cache, birds cache, humans cache. So we can look across all these different species at cache size. Some species have large cache sizes, some species have small cache sizes, some species have medium cache sizes. We wouldn't want to put these kinds of quantities, but we might quantify it in terms of the number of calories. Large caches have lots of calories, and we could actually put a number on that. Small have very few calories stored, and other ones have, a medium number of calories stored. And then within this framework of quantifying the cache in terms of calories, we could also say, well, what is the level of polygyny? Well, the level of polygyny might be the deviation from monogamy. So monogamy would be a 1-to-1, and so a 1-to-2 male to female ratio would be somewhat of a deviation. A 1-to-12 would be a more significant deviation from monogamy. And maybe this person has the hypothesis that cache size influences level of polygyny such that individuals with large caches tend to have a further deviation from monogamy than individuals with small caches. Something like this. So we could build a data set where we quantified the deviation from monogamy by just dividing 1 by 12, 1 by 1, 1 by 6. 1 would be monogamy. As the number gets smaller, you're further and further from polygyny. And then I could say, okay, how would you test this idea? Would you test it by looking at individuals within a species where there's inevitably variation in cache size, and there's also probably some variation in degree of monogamy? Would you look across species and do this? And what would be the advantages and disadvantages of those two ways of doing this? So would you take a single species approach? inter-individual variation? Or would you take a multi-species approach? Where you look at phylogenetically independent contrasts. Which would you advocate in this case? Think about humans. Do humans have different caching tendencies, probably? Do humans exhibit differences in degree of polygyny? Probably. Do you know of many humans where they have 1 male to 12 females? No, you'll oftentimes have 1 male to 2 females. If you really have a lot of time on your hand, maybe 1 male with 3 females, 1 male with 4 females, that would be a lot to juggle. But you're not going to have as wide a variation with looking within humans, for example, probably in either cache size or levels of polygyny. If you are now looking at humans versus squirrels versus shrikes versus other primates, etc., would you expect to see more variation in cache size or less variation in cache size than looking at a single species? What?
You would expect to see more variation.
You would likely see more variation. What about levels of polygyny?
You'll also see more variation.
More variation. So of those two approaches, which would you probably want to be using?
Compare species.
You would probably want to look among species, but then when you looked among species, you wouldn't want to just take the raw data. You would actually want to look at phylogenetically independent contrasts to remove any effect of phylogeny that there was there. That's a fair question to ask. Now, I realize that question required a lot of preamble, and I'm known to have questions that have a certain amount of preamble on classes, but that's the kind of question that I might ask. I might give you a data set that has a graph that shows the relationship between trait X and trait Y across a variety of taxa, where it's just the raw data, and then give you another plot that is the relationship between X and Y after controlling for phylogenetically independent contrasts, and ask you, are these two traits correlated with one another? As trait X varies, does trait Y also vary based on those two figures? And if they're correlated in one panel and they're also correlated in the other panel, then there's a good, strong evidence that those two things are correlated, meaning that they're driven by the ecological circumstances of those species, because the phylogenetically independent contrasts still maintain that correlation. But if they're correlated in the single taxon case and they're not correlated in the other one, then what's really driving that initial correlation is the fact that phylogenetically related species are tending to do similar things because of their shared evolutionary history, because when you remove that evolutionary history from the equation, those two traits are no longer correlated with one another. That's another kind of scenario that I could invent and give you. Does that make sense? Phylogenetically independent contrasts are hard to ask test questions about, because really the way to really see if you understand it is to actually do it, and we're not going to take the time to go and do that. You're going to see throughout the textbook that in some cases they offer you case studies and examples where they're looking at variation among individuals. In other cases, they show you examples where they look at how a particular set of behaviors vary across broader phylogenetic scope. And in those cases, a lot of times the studies that they will talk about are these comparative analyses that look at phylogenetically independent contrasts. So you need to be aware of it way more than you need to be able to recreate it. Does that make sense? Okay. So there are limits of the comparative method. One limit is that there's oftentimes not enough attention paid to alternative hypotheses, because in general, the researcher is interested in how trait X relates to trait Y. And because they're interested in how trait X relates to trait Y, they oftentimes don't think about trait Z. And so just the act of engaging in a comparative method can canalize your thinking to other potential alternatives. Oftentimes, ecological variables are not quantified very well. So, if those ecological variables are important to the evolution of those traits, then having more information about those things is better than having less. Even after you do the comparative method, correlation never means causation. Unless you can experimentally alter the X variable and see how the Y variable responds to that alteration or manipulation of X, you can't actually assign causation. So even a well-done comparative method suffers from the fact that all you can say is that these two traits covary with one another. One is not necessarily driving the other. And then we also sometimes see non-adaptive behavior. One of the assumptions of the comparative method is that evolution, natural selection, is optimizing behavior so that behavior is adaptive. And sometimes we see what we interpret as non-adaptive behavior, and we struggle to explain it. And so oftentimes we avoid that behavior. But then the other thing is that there's this idea in ecology and evolution that there might be multiple adaptive peaks. There might be multiple solutions to a common problem, and those multiple solutions are all equally valuable. And it just depends kind of on the individual trajectory of how something evolved in a group of organisms as to which of those alternative adaptive peaks you ended up on. So lots of solutions to the same challenge. The question of which solution your particular species arrived at is all a matter of where you started and how you got there, rather than that's the best solution. There are multiple solutions that are all equally good. And then this problem that just phylogenetically species are not statistically independent. This is a more kind of purist statistical way of thinking about this. Presumably, the reason that we use phylogenetically independent contrast is it gets rid of this particular issue. Phylogenetically independent contrast is designed to deal with this last limitation. Phylogenetically independent contrast does not deal with any of the earlier limitations of the comparative method. So, we looked at this last time. This was a form of constraint. In this case, the researchers of this study looked at how the size of offspring affected the number of offspring that individuals had. This is your classic ecological size number trade-off. As you make bigger babies, you have to make fewer of them. If you want to make more babies, you have to make each baby smaller, because you only have a set finite box of energy to devote to reproduction. Now, these things are negatively correlated because of this size number trade-off. But this figure here, each of these dots is a taxon. Humans are here. These are not phylogenetically independent contrasts. So here you have the great apes. Here you have the non-ape primates, and you see that these two things have similar slopes, but they have different y-intercepts. That difference in the y-intercept is probably not due to the inherent nature of the size number trade-off. That probably has to do with the fact that this line here is one phylogenetically related group of individuals, a group of taxa, and this line here is another phylogenetically related group of taxa. This line, the lower line, corresponds to this purple group of taxa. This upper line here applies to this group of taxa. And so, while there still may be a negative correlation between the number of offspring that you have on the y-axis and the size of offspring on the x-axis, this particular figure probably has a certain amount of phylogenetic signal in the data, because these are the raw data from the taxa. These are not the phylogenetically independent contrasts, and that's probably why this line is different from this line. If we were to do phylogenetically independent contrasts on this data set, we should expect that difference between the haplorhines and the great apes to go away, to evaporate, because we will have removed the effect of that shared phylogeny from the data set. I don't know why they didn't do phylogenetically independent contrasts. They've been around long enough, but they could have in this case, and they, for whatever reason, didn't. And they still got it published in the Proceedings of the Royal Society of London, which is a pretty good publication. Oh, I'm sorry, I just stole my thunder. This is, I forgot about this. I forgot about the last slide, because I guess I didn't prepare well enough. This is one of the figures in this paper showing you just the raw data. This is the phylogenetically independent contrast. And so, once again, you do phylogenetically independent contrasts, and the thing that I said would happen actually happened. That distinction between the great apes and the haplorhines disappears. And so this would be a truer measure of the strength of this trade-off between the size of offspring and the number of offspring being produced. Stole my thunder. That's all I have. Questions?
Can you go back to slide 48?
I don't know. What is slide 48?
I don't know. I was looking at your presentation because I was taking notes on it.
Great. Oh, this one?
Yeah. So I know like the difference between this and the one where they have no correlation is because you remove the phylum. But how do you do that?
So in this case, what you have is you have this phylogeny. And so if you zoom in on this phylogeny to this thing right here, in this figure, you have a certain number of points, and each of these points refers to one of these terminal taxa, one twig at the very tip of this. What you would do to do phylogenetically independent contrast is you would ask yourself, for trait 1 and trait 2, what is the difference between trait 1 and trait 2 in this taxon and that taxon? That would be your contrast.
Okay.
What's the difference between this taxon and that taxon? That's your contrast. So, in the same way here, where did I go? You would look at the difference between trait D and taxon D and E for trait 1 and trait 2. The difference here is a difference of 4. The difference here is a difference of 10. That's contrast 1. Difference of 4 for the X character, a difference in 10 for the Y character. And so that tells you, over this course of evolutionary time, how much did trait X evolve and in what direction, and how much did trait Y evolve and in what direction. And if X and Y are related to one another because X is an ecological driver of Y, then those two things are evolving in concert with one another, or y is evolving in response to x. If that's the case, that the environmental driver is affecting a morphological characteristic, for example, then that degree of change over that degree of evolution should be similar everywhere. The longer two groups have been apart, the more change you should expect to see, but you should expect to see it going in the same direction if the ecological driver is also driving the morphology. So here, we might expect to see similar levels of divergence, 4 to 6, which is 2, 87 to 93, which is 7, 87 to 93, which is 6. Once again, the two seem to be moving in concert with one another. These two groups are separated by a much broader, a much longer evolutionary history. As a result, you might expect their divergence to be greater. But once again, if these two things are correlated, if the ecological driver is driving the evolution of the morphology, you should once again expect those things to be moving in the same direction as everybody else is. They are just moving for a longer period of time because there's a longer evolutionary history separating them. Cumulative time on this arm plus the cumulative time on this arm. And what we find is that they are different, and they're different for the magnitude of the differences are bigger, mainly because they have been separated for a longer period of evolutionary time. So when you look at,
So would that one be a positive correlation then? I mean,
that is a
assuming-
positive correlation, assuming that you had a bigger phylogeny that you did this across. So what this method was, I basically generated the phylogeny, and then I told R to do a random simulation of phenotypic evolution on this phylogeny. For these time periods, a long divergence followed by a rapid evolution. Long divergence followed by a rapid evolution. And I said, do this for two traits, trait X and trait Y, but it's random. It's just random. Make it jigger around, make it flow in a tree, but the direction of flow is random for X, and also it's completely independent and random for Y. So that it looks like this random movement of phenotype. We get to the phenotypes at the endpoints. Those are the phenotypes of these terminal taxa. And then we just look at the raw data. We look at X and we look at Y. And what we find is that X and Y look correlated. But they look correlated because they're these two portions of the phylogenetic tree.
So those values are like the raw. Like
These are the
individual, like,
these are the individual values at each of these points.
Okay.
So there should be as many points on this figure as there are terminal taxa on this tree. And so this upper group of points here is all these taxa in this part of the tree. The lower one is all of these taxa down here. So it isn't that you have a bunch of independent points.
Mm-hmm.
You have one point here, and you have another point here. And you draw a straight line between 2 points, and you get a straight line. So they're not really independent. They cluster together because they share a common evolutionary history. So the attempt to get out of that is to, instead of looking at the actual raw data, look at these contrasts. These contrasts look to see if evolution in the two, or change in the two traits, is actually correlated with one another or not by comparing sister taxa at the very terminal ends, looking at deeper levels where you compare this common ancestor to this common ancestor, this common ancestor to that common ancestor. So instead of having 1, 2, 3, 4, however many terminal taxa in the next graph, you have the number of points that correspond to all these independent contrasts. And when you do that, because the data that I put into the analysis were generated at random, you shouldn't actually expect to see any correlation because the evolutionary trajectory of trait X was random and independent of the evolutionary trajectory of Y. So if phylogenetically independent contrast actually gets rid of this effect of phylogeny, what we should see is no pattern at all. And that is exactly what we see. Okay? So that was kind of the point of all of that.
Okay.
Is that clearer?
Yeah.
now?
I think I just
Good.
missed how we got the first few points.
Okay. All right.
Do either of these have to deal with heritability and convergent evolution? Like,
What's that?
Like, do either of these have to, like, can either of these tell you that, like, it's convergent evolution or inheritability, or is that just not something we can determine?
Yeah, what you would find if you had traits that were convergent is they would be in this cluster of points. But once again, if the overall pattern is patterns of convergence, it would depend on how much convergence is actually going on. If there's a lot of convergence going on where lots of different taxa are responding to the same environmental drivers in similar ways, then you would see a correlation. That would be a real correlation. And because it's convergent, it already started out being phylogenetically independent because you have one thing arising in one lineage and that same thing arising in another lineage. So in that case, you would get a situation where the raw data would show the correlation, and also the phylogenetically independent contrast would also show the correlation. What we're trying to avoid with phylogenetically independent contrast is a false positive, finding a correlation that isn't actually true. You can do the raw data and then do the phylogenetically independent contrast, and if the correlation is in both those cases, then that correlation is real. If it's true in the raw data, but it's not found in the phylogenetically corrected data, then that original phenotypic correlation wasn't really a real thing. It's just an artifact of the fact that those species are related to one another. And you should always be doing things in a way as a scientist that give you the true answer rather than a false answer. You're trying to avoid a type 1 error, saying a correlation exists when it doesn't. Okay? Type 1 error. Avery took stats last year. Other questions? Well, good, because we're done. All right. Email me if we're getting together tomorrow. If we don't get together, you can just slide your papers underneath my door, and I'm going to pick them up on Friday.
Yeah, I'd really like to come in and do the review during my time if you want to.
What's that?
I would come in.
I'll come in and do the review tomorrow.
Okay, then I'll see you guys, one or more of you, tomorrow at the beginning of lab. We'll just hang around as long as you need to. I would normally go into the field, but it's going to be bitterly cold, and I know because my wife's family lives in Smithville and we go up there fairly often. They're always 2 days behind everybody else on clearing snow, and there's no sense in risking life and limb to observe birds. We'll do it the next week. Like I said in my email, there's some wiggle room built into the schedule, so.
I'm going to be on the road Thursday. If you want me just to email you my lab report?
Sure.